Endocrine response to resistance exercise. The endocrine system supports the normal homeostatic function of the human body and helps it respond to external stimulus. Its importance in the field of strength conditioning is reflected by the critical role it played in the theoretical development of periodization of training. Hans Sale, a Canadian endocrinologist, unknowingly provided the theoretical basis for periodization with his work on the adrenal gland and the role of the stress hormones in the adaptation of stress, distress, illness, and death of an organism. Former Eastern Bloc sports scientists and physicians found similarities between the pattern of training response in athletes and the stress pattern observed by Sale. Hans Sale coined the term general adaptation syndrome to describe how the adrenal gland responds with an initial alarm reaction followed by the reduction of an organism's function in response to a noxious stimulus. And the stimulus does not kill the organism. This amazingly is followed by a period of adaptation or improvement in the organism's function for a given period of time. This improvement then plateaus at this point. If the noxious stimulus is not removed, the subsequent decline in function sometimes leads to the death of the organism. The key to continued adaptation of the stress is the timely removal of the stimulus so that the organism's function can recover. It is important for strength conditioning professionals to have a basic understanding of the hormonal response to resistance exercise. Such knowledge provides greater insight into how an exercise prescription can enable hormones to mediate optimal adaptations to resistance training. Although resistance training is the only natural stimulus that causes increases in lean tissue mass, dramatic differences exist among resistance training programs in their ability to produce increases in muscle and connective tissue size. The type of resistance training workout used dictates the hormonal response. Tissue adaptations are influenced by the changes in circulating hormonal concentrations following exercise. Thus, understanding the natural anabolic activity that takes place in the athlete's body is fundamental to successful recovery, adaptation, program design, training progression, and ultimately athletic performance. Synthesis, storage, and secretion of hormones. Hormones are chemical messengers that are synthesized, stored in, and released in the body by endocrine glands, body structures specialized for secretion, and certain other cells. Similarly, neurons synthesize, store, and secrete neurotransmitters, which may also have hormonal functions. The relatively new term neuroendocrinology describes the close relationship of chemical substances that have both neural and hormonal functions. Typically, endocrine glands are stimulated to increase or release hormones by a chemical signal received by receptors on the gland or by neural stimulation. Excuse me. For example, the adrenal medulla, the internal part of the adrenal gland, releases the hormone epinephrine or neural stimulation from the brain. The adrenal cortex, the outer part of the adrenal gland, synthesizes and secretes the hormone cortisol. After stimulation by another hormone, adrenocorticotropic hormone, ACTH, released from the pituitary gland. Following stimulation, endocrine glands release hormones into the blood, the basic definition of a hormone, which carries the information to hormone-specific receptors on target tissues, peptide hormones, or directly to the DNA in the nucleus of the cell, steroid hormones. In addition to endocrine release into blood circulation, hormones can also be secreted by autocrine and paracrine mechanisms. Autocrine secretion of a hormone means the cell releases the hormone inside itself. It may stimulate to do so via an external stimulus, another hormone, but the secreted hormone never leaves the cell that produced it. For example, insulin-like growth factor, IGF-1, could be released inside a muscle fiber on the stimulation of a mechanical force produced due to the growth hormone interactions with the muscle. Paracrine secretion of hormone involved the release of a hormone to interact with adjacent cells, without the need for circulation to put the hormone in contact with the target cell. These mechanisms demonstrate the multi-role that hormones can play in their interaction with a target cell. A variety of binding proteins that carry hormones are found in the blood. These many different binding proteins carry both peptide hormones and steroid hormones. In a sense, these binding proteins act as a storage site within the circulation, help fight degradation of the hormone, and extend its half-life. Most hormones are not active unless they are separated from the specific binding protein. However, some hormone binding proteins themselves may actually have a biological action. Thus, binding proteins themselves may actually have biological actions. Thus, binding proteins, whether circulated in the blood or bound to a cell, are major players in endocrine function and can be thought as a type of receptor in some cases. Growth hormone binding proteins. Many hormones affect multiple tissues in the body. Testosterone, or one of its many forms, for example, interacts with almost every tissue in the body. In this chapter, we focus on the skeletal muscle tissue as a primary target of hormonal interactions, but many other tissues such as bone, kidney, and liver are just as important as muscle to the adaptive changes observed with resistance training. Most hormones play multiple physiological roles. These roles include regulating reproduction, maintenance of the external environment, energy production, utilization and storage, and growth and development. In addition, hormone interaction in complex ways with each other. A particular hormone may function in either an independent or dependent manner, depending on its role in a given physiological mechanism. This complexity and flexibility allows for the endocrine system to respond to the proper magnitude of the physiological challenge and to interact differently with each physiological system or target tissue simultaneously. Muscle as the target for hormone interactions. Skeletal muscle is somewhat unusual in that it has multinucleated cells. This means that the protein within a muscle fiber is controlled by multiple nuclei. Nuclear domains are important in these cells and make possible differentiation and regulation of protein metabolism along the length of the muscle fiber. This can be observed in the changes in the diameter of a long muscle fiber. Muscle fibers are usually no longer than 4 to 5 inches, or 10 to 13 centimeters, down the length of the fiber, which result from differential control of the protein by different nuclear domains. Hormonal mechanisms that interact with skeletal muscle are a part of an integral system that mediates the changes made in the metabolic and cellular processes of muscles as a result of the resistance, exercise, and training. Muscle remodeling involves the disruption and damage of muscle fibers, an inflammatory response, hormonal interactions, and ultimately the synthesis of new proteins and their orderly incorporation into existing or new sarcomeres. The inflammatory process involves the immune system and various immune cells, T cells, which are under endocrine control. The connection between neural, endocrine, and immune system is called neuroendocrine immunology. The term demonstrates the interdependence of these systems and the integrative nature of the remodeling process in muscles. We cannot limit our thinking about biological processes in our system. The most prominent resistance training adaptations in muscles is an increase in the amount of muscle contractile proteins actin and myosin. Other changes in these proteins are also significant. For example, heavy chain myosin proteins can go through a change in their molecular structure from type 2B to 2A, heavy chain proteins. More important, the synthesis of non-contractile proteins is needed for structural integrity and orientation of the contractile proteins within the sarcomere. The non-contractile proteins are laid down first in a coordinated physiological development of muscle size. Stimulation of protein synthesis by heavy resistance training starts at the level of the gene. It allows both the quality and quantity of muscles to be altered over a period of time. The hormones are involved in the ongoing process of development. In general, the increase in the protein synthesis and decrease in the protein degradation are the first steps in muscle growth. However, this differs between muscle fiber types 1 fibers dependent more on the reduction of protein degradation, whereas type 2 fibers depend on a dramatic increase in protein synthesis to maintain their size or hypertrophy. The difference significance influences the role that hormones play in differential regulation of protein metabolism in type 1 and type 2 muscle fibers. Hormones are intimately involved with protein synthesis and degradation. Mechanisms. The production of the contractile proteins actin and myosin, and ultimately their incorporation into the sarcomere, complete the process of the molecular level. A multitude of hormones, including anabolic hormones, hormones that promote tissue building such as insulin, insulin-like growth factor, testosterone, and growth hormone, all contribute to various aspects of this process. Thyroid hormones act as an important permissive hormone that allow for actions of other hormones to take place. As another important action in the building of tissue, anabolic hormones also block the negative effects of protein metabolism of catabolic hormones such as cortisol, progesterone, which attempt to degrade cell proteins to support glucose synthesis. The remodeling of muscle fibers involve changes in protein metabolism, the structure alterations and additions to take place during the recovery period of an exercise stress. The more muscle fibers recruited for a performance of the exercise, the greater the extent of the potential remodeling of the whole muscle. The relationship among hormone muscle fibers and the subsequent changes in function capabilities of muscle fibers provide the basis for the adaptive influence of hormone in hypertrophy. The roles of receptors in mediating hormonal changes. Excuse me again. Ultimately, the hormone is trying to influence the DNA to the nucleus of the cell. Yet direct receptors for all hormones are not found on the DNA. Such hormones have to influence the DNA via secondary reactions. Receptors are found in a mobile form, binding proteins for growth hormone, integrated into the cell membrane polypeptide receptors, and on the regulatory elements of the DNA, steroid receptors. Every cell, from muscle fibers to brain cells, have receptors to mediate the message of the signal form hormone. One of the basic principles of endocrinology is that given hormone interactions with a specific receptor, a phenomenon known as the lock and key theory, where the receptor is lock and the hormone is the key, although only one hormone has exactly the right characteristics to interact with a specific receptor, the cases of cross-reactivity, a certain receptor partially interacts with a hormone that is not specifically designed for, allosteric binding. When this occurs, the resulting biological actions are different from those induced by the primary hormone. Receptors can have allosteric binding sites at which the subsequent substances other than hormones can enhance or reduce the cellular response to the primary hormone. Receptors may also have a number of domains. This means that the receptors may outside the cell membrane, externally within the cell membrane, internal, or part inside and part outside the membrane, integrated. Growth hormone, a polypeptide hormone, is an example in which the receptor takes on several of these domains. Steroid receptors are situated on the nuclear regulatory protein of the DNA, of the cell's DNA. It is usually the hormone receptor complex that transmits the message to the nucleus of the cell. The genetic material within the nucleus ultimately translates the hormonal message into either inhibition or facilitation of protein synthesis. When an adaptation is no longer possible, maximum amount of proteins have been added to muscle fiber, receptors can become non-responsive to a specific hormone's message and may be trying to stimulate the protein synthesis response in the cell. This inability of a hormone to interact with this receptor is called downregulation, or receptor function. Receptors have the ability to increase or decrease their binding sensitivity, and the actual number of receptors present to bind with can be altered. Alterations in the receptor's binding characteristics or the number of receptors can be dramatic in adaptation as the release of an increased amount of hormone from an endocrine gland. Obviously, if a receptor is not responsive to the hormone, little or no alteration in the cell metabolism will result. For example, it has been shown that hormone testosterone that exercise training affects only the maximum number of binding sites, but not the binding sensitivity of the receptors. Scientists are just starting to study and understanding the role of receptors in muscles during exercise training.